are made, we'll make them available to everybody and so forth. All right, so I left with this boring slide yesterday. And this just touches on a number of features that we quickly went through in the context of how do we generate this level of diversity in the antibody molecule that I've been talking about. So went through those mechanisms pretty fast, just in probably 20, 30 minutes to talk about mechanisms of recombination, the role of RAG proteins, the P and N nucleotide story, contributing to diversity and so forth. And so it is, we do expect that you remember those items, and some of the key things I've listed here that I want you to remember. This is a big issue here, that a B cell makes an antibody of a single specificity. So one cell is not producing multiple different antibodies of different binding specificities. They're producing a single antibody of a defined specificity. A lot of times people forget when we're talking about that level of diversity and the variable region, that binding pocket, the mirror image of antigen. Remember, both the heavy and the heavy chain, we're generating a level of diversity. We're getting rearrangements on the light chain, and then when those come together, that combination is contributing to a level of diversity. Also remember this concept of allelic exclusion. So at the end of the day, we are only going to use a successful recombination event on one of the copies of our genes. And again, this ensures that a B cell produces an antibody of a single specificity. Again, combination light and heavy chains are contributing to the specificity. These rearrangement events that we quickly went through yesterday, we have cells that are destined to be a B cell. And the development of that B cell, and for simplicity, we're just going to be using the bone marrow as our site of development. When I move maybe this morning into our development lecture, we're going to be talking about how when the B cell is going through development, learning to be a B cell, so it's an immature cell at this point, that's where these rearrangement events are occurring. And they're tightly controlled. And they will define steps of development. Okay? And so I'll be coming back to these recombination events as we bring you through the development of a B cell from a progenitor to what we're going to define as a mature but naive B cell. Naive meaning it hasn't seen foreign antigen. But it'll be a mature cell, and that's going to be defined by the surface of expression of both IgM and IgD on the surface of the cell. And so yesterday I talked about the fact that once we get successful recombination, we get transcription through the constant genes, and we transcribe both IgM and IgD into the same transcript, and then RNA splicing mechanisms generates an IgM specific and an IgD specific, so we can make those proteins. And they both go to the surface. This is what we are going to use as our markers for mature yet naive B cells. It's a diagnostic tool for you as you're looking at reasons why your patient is having trouble with extracellular pathogens, and whether or not you are even generating the population of B cells. That IgM that's produced on the surface, it can be secreted. IgD is rarely secreted. I mean, you can find a little. IgM is important. It's our first antibody we're going to make, first soluble antibody when we engage a pathogen. IgD still has poorly defined functions. And I think I mentioned yesterday, it's still conserved in all animals, so by definition it must be doing something important, but this is one that we do a lot of hand waving on, and I'll give you a little hand waving of the potential function of IgD when we talk about the development of B cells and so forth. The rest of those classes of antibodies can be made as a membrane form and a secreted form. So it's only IgD that's a little weird. And then also remember, for simplicity, we talk about B cell, B cell, B cell. We're getting surface Ig. But if we're going to get large amounts of secreted antibody, that B cell has to differentiate. And it differentiates into this plasma cell. And this plasma cell is going to devote most of its energy then to the production of a secretory antibody. And one of your diagnostics tools for looking at whether or not your patient has frequency of plasma cells is actually the loss of surface Ig. And that's a key feature of a fully differentiated plasma cell. So those are just a few key bullets that kind of touch upon some of the things that we talked about yesterday. Now, when we're talking about that diversity, yesterday we talked about generation of diversity before we ever see antigen. So we're doing this in development. Our B cells are coming up in the bone marrow, and we're rearranging genes, and then we put out into the periphery this incredible repertoire of Ig molecules that are expressed by these B cells that go out, and now those B cells are going to start recirculating through our secondary lymphoid organs, and if they ever see antigen in the future, then they're going to do their job. So we define this diversity before antigen, and that's that recombination events that we talked about yesterday. It is completely random, the way those recombination events occur. And we're just generating incredible repertoire of cells. Some of those cells, in fact, a lot of those cells, will never see antigen in their life. You know, we do generate a lot of B cells in this setting that have surface Igs, fully functional surface Igs, but in the life of that B cell, it will never see an antigen that'll stimulate it, and so forth. However, we do stimulate a lot of our B cells as we encounter foreign antigen. So recombination generates diversity, and then that junctional diversity that contributes to basically the generation of new exons, those P and N nucleotides. So P for palindrome, and look at that cartoon, how when you cut it and fold it up, and then you fill in, the sequence going backwards is the same as the sequence going forwards, palindrome. And then the random nucleotides that are put at the end, and so I guess we couldn't spell, N is for end, and that's generating diversity. And so remember the CDRs, and I'll come back to the CDRs later, but the CDRs are those loops that actually make the binding pocket. Which of the CDRs are junctional diversity generating the greatest amount of variability? 3. And that's simply because of the location. Back at the end, yeah.
Are those introducing multiples of 3, or are we creating a fraction?
Excuse me?
Those nucleotides?
Yeah.
Are they introduced in multiples of 3, or are we creating a fraction?
They're not in multiples of 3. That's for simplicity in the cartoon. Okay? So you just need to take from it that we have the addition of these nucleotides. Those cartoons that are in the book that I use and so forth oversimplify. Okay? Okay, so this all takes place before, then we're bringing together a heavy and light chain, like I mentioned up here. And then we produce a transcript with the IgM, IgD, and then we splice it. And we make an IgM and IgD protein that's out on the surface. So that's all done. Now we got our mature naive cell. Gets exported to the periphery. When we talk about development, I'll tell you how we get rid of those B cells that are autoreactive. Because this is truly, truly random. We have just as good of a chance in this process to generate a B cell that has a surface Ig that sees us as we do that sees something foreign. And so they are generated. So what we have to do is get rid of those. And we do it a couple different ways. One of the ways is we'll kill it if it's autoreactive. And then other ways we'll make it, we're going to introduce this term anergic, so it's non-reactive. And we'll make some of them anergic. And for B cells, they do have kind of a cool feature that we'll mention, that if we didn't make a good one, we can go back and try it again and rescue it and so forth. And we'll talk about that in our development lecture. Now, after antigen, what's going to happen that might contribute to this diversity response? All right, yesterday I talked about the fact that antibodies can improve. They improve after antigen. Okay? So we first make an IgM antibody, and most of the time that's not sufficient. We want to switch the class of the antibody or the isotype of the antibody. So one of the ways that we change things after antigen is change the class, change the functional properties of the antibody. The other thing we do is we improve the affinity of the antibody. So I mentioned that yesterday. Affinity maturation. And we actually will mutate the binding sites, and we'll get to that in the next slide here. And then the other thing that we're going to be doing when that cell transitions to a plasma cell is that instead of wasting energy making a membrane form, we're going to make sure everything is a secreted form. Same starting transcript, and we do that with a splicing mechanism as well. All right, and so the difference between a surface IG and secreted, same antibody, let's say it's an IgM or an IgG, that's a membrane form. It has a transmembrane domain. We simply splice that out, and now that thing won't get hung up in the membrane and will get secreted. And that plasma cell is going to do that, and it's an impressive factory for secreted protein. All right, this is the thing that I think is just absolutely wild as well. So we randomly generate these B cells. We export them to the periphery. They're floating around. Now you've breathed in your rhinovirus. And we have B cells that engage, that respond to antigens from that virus. And we'll expand. We'll get plasma cells. They'll make that IgM antibody. It'll go back and try to get rid of the virus, but many times doesn't, and could be the class of the antibody. But it also could be the fact that that IgM is just too low affinity. It's just not binding tight enough, so the on-off rate is such that it just isn't sufficient for neutralization of the virus or maybe clearance of the virus. So that B cell that originally responded, it would have responded in a secondary lymphoid organ, so a lymph node. So it's responded to antigen. Remember clonal proliferation. We've generated lots of progeny. Some of them become plasma cells. They're making antibody. That ain't working. And so the process is continuing because we're not getting rid of the antigen. So the antigen is still there. It's still being presented. The B cells are there. We've got T cells that we haven't brought into the picture yet other than in my overview lecture. But we've got these T cells called T helper cells. They are also responding to the foreign antigen, but as I introduced in the overview, it's not responding to the same epitope that the antibody, the surface IG is, to the same antigen, but a fragment. We generate these T cells, and they're going to provide help to the B cell. And they will provide this help, particularly in these cases where we're not getting rid of the antigen. So the T cells are being expanded, activated, and they are now interacting with the B cells that have responded to antigen. And we will get a class switch. The IgM is going to be switching to an IgG most of the time in this setting. That's going to improve the function of the antibody. That's a good thing. But then there's the other thing that's occurring that I've referred to as affinity maturation. And that is when we look at the nucleotides within these CDRs during the course of an immune response, as we're trying to get rid of antigen, and these little red spots are changes in nucleotides that are due to a mutation event. And what we get are random mutations in the CDRs. This is taking place in that follicle within the germinal center, that activated site. It is controlled by the T cell. The T helper cell, one of its things that it's doing to the B cell is, and I'm going to introduce another cytokine here. We have a lot of cytokines in immunology that I could talk about, but I keep it pretty restricted. And it's kind of my bias, which are the most important. I picked the ones that help define the biology of what we're talking about. So this T cell is providing help. It provides help by cell contact, and I think I had a cartoon in my overview of that. But in that cartoon, if you went back and looked at it, I had a little arrow of factors that are helping. The T helper cell that's relevant here, that is providing help, is making a factor called IL-4. This IL-4 is critically important in this B cell improvement, or the antibody improvement that's going to take place, along with some other cytokines in that cell contact event. But right now, really remember this IL-4. This is the one that really is distinguishing this response, and it's controlling this, or contributing to the control of this environment now this B cell is in. There will be enzyme systems that are activated and so forth, and there's this guy, activation-induced cytidine deaminase, or AID, and this is what's controlling the mutation events. So pretty wild that we are, because normally when you're talking about mutating DNA, that's not normally a good thing, and so forth. Here, the immune system is using it to its advantage. It's going into these sequences, and so this is an antibody that just didn't work, and we're randomly mutating nucleotides in these sites to where the longer the antigen stays there, the more mutations we're going to see. Now, these mutations are random. We have just as good of a chance of making the antibody worse as we do making it better. So some of the mutations are actually going to make the antibody bind less to the antigen. However, some of the other mutations have the potential of making the antibody binding better. So what happens is, everything is still being driven by antigen. The cells that are responding, the B cells that are responding. If their surface IG gets worse, they quit responding. If their surface IG gets better, they continue to respond. And then as antigen level goes down, because hopefully we are making antibodies now that are starting to clear it, the antibodies of the highest affinity, surface IG of the highest affinity, will be the ones that will be activated the longest. Right? Because antigen level goes down, the receptor with the highest affinity is going to continue to be stimulated. So at the end of the day, what's happening here is we're slowly developing antibody responses that are clearing the bug. As the bug is disappearing, the antigen's disappearing, we are preferentially expanding the best of the best of the B cells, those with the highest affinity. And they continue to expand. And during that process, we're going to be generating some very, very long-lived cells that we call those memory cells that we'll talk about. So this affinity maturation process is really, really important. And it's controlled by this AID for an enzyme. It's regulated by the T helper cell, and one of the key factors is this IL-4. Cell contact is also absolutely critical. So, if your patient lacked AID, okay, what would be the phenotype in that patient? And this does happen. So what would be a diagnostic readout that you would get from, because you have suspected there is some immunodeficiency in your patient. So you sent their cells down to the lab, came back, and they're not going to tell you it's AID deficient, but let's say your patient is lacking a functional AID, what would be the phenotype? Yeah.
Homogeneous.
What's that?
The B cells would be homogeneous genetically, you wouldn't have any mutations. Or
You
less-
wouldn't see the mutations. It wouldn't be homogeneous because they all still have generated a different IG molecule. The diagnostic lab is not going to give you the mutation analysis. You'll be able to do that later on, but your initial diagnostic tests would not give you mutations. Yeah.
Would it, you would have normal levels of the cells, but they would be responding to the bug? It wouldn't be clear here?
You could have normal levels of the cells. You aren't clearing the bug. This is what has now got you thinking, all right, we've got a deficiency here. All right, you've already have gathered data because you're getting it from this class and so forth, that your patient is coming back with extracellular infections. And you treat with antibiotics and you're clearing and they're coming back and so forth. And so you're sending down, you're suspecting now immunodeficiency. And you don't know it yet, but your patient lacks AID. You guys are getting close.
They've already made the IgG, right? There's already been that switch before AI?
No.
So then you can just look for that.
You'll see the early response taking place, and we're going to see IgM in your patient. And we're going to get back to the mechanisms of this particular disorder when I get to my T cell here. So I kind of jumped ahead in that we're going to be talking about a hyper IgM syndrome in that setting. All right, anyway, so laundry list again. Sorry about that, but so antibodies with effector functions are generated by our isotype switching. And we've already introduced our different classes. It does get more complicated that within the IgG class of antibodies, you have subclasses. In the early years I taught this course, I would tell the students, well, right now just think about the major classes. But amazingly, frequencies of these subclasses are diagnostic. And that these subclasses of IgG do have some subtle differences in what they do, but frequency of these end up being a diagnostic tool for you as well. You're going to have an immunodeficiency disorder that I think would be really, really hard to diagnose. It's called common immuno... Now I'm blanking out on common... Well,
Skid.
not SCID. No, this is... Come on, Diane, help me here. Common, it's IG deficiency or something like that. It's basically a subtle deficiency in some isotypes of antibody. And this deficiency can present late in life. A person could be in their 40s. And they're starting to get infections in the lungs that they really can't clear. And it ends up being a very subtle effect on some of the isotypes in the patient. And the treatment in that setting is just IG replacement. Very unusual immunodeficiency. And again, it'll come from the analysis of the subclasses. Anyway, back to our bullets. We talked about antibodies as neutralizing or opsonizing. IgMs are the first antibodies that are made. All antibodies are bivalent, but IgMs, we have this ability to put 5 of them together and increase their avidity. Yeah?
During opsonization, do the macrophages recycle, or are the antibodies recycled once they're taken in by the neutrophils?
Are the antibodies recycled when the antibodies go in? Antibodies are very resistant. And if they aren't degraded and their antigen dissociates and so forth, there would be the potential. But I would think it's a pretty minimal impact on antibody levels because the antibody has bound up its antigen. The cell is taking in that bacteria. It's chopping it up and so forth. The antibody is being exposed to all the harsh enzymes and so forth, though it is very resistant, like I said. And I would think at the end of the day, the bulk of it would be destroyed. But I bet there are situations where it's not. And, you know, the neutrophil and the macrophage, one of the key functions of regulation, particularly of the neutrophil, is that we want it to die. And so if it's still there, functional, it'll get released, but I would think it would be an incremental impact. That would be my prediction on that. It would be incremental.
I'd agree on that.
Yeah.
I mean, there's phagocytes that's there efficient in breaking down what they take out because that's their job.
That's their job. Yep. Okay, so opsonized IgM, somatic mutations, we increase affinity. IgG, we'll get a class switch. It's our most abundant antibody in the internal fluids. How's that? It's our most flexible antibody as far as function and so forth. We have lower levels of IgA found in circulation. It's intended to protect basically outside the body, that secretory function. And I've mentioned IgEs just briefly so far, but it's another class of antibodies that we can switch to. And where I have brought it up is that when you guys see it, it's going to be in the context of most likely allergic responses and so forth. But we feel that it has an important role in expulsion of parasites. And it's kind of a hand-waving thing because a lot of times, well, when I've taught this question in this class in the past, a common question is, why do we have IgEs? You know, if they generate this weird pathology and so forth. Well, where our hand-waving goes, and it's really tough to prove in humans, is that they are important in responses against certain parasites and so forth. And it was a very effective mechanism. And one of them is, how do you deal with things outside the body, so within the lumen of the gut, that IgA may not be dealing with and so forth, and these large intestinal worms or things along those lines that are outside the body, how do you deal with them? And you'll see these in your path lectures and so forth, pictures of helminths, certain parasites that gain access into the surface tissue and so forth, and accumulation of eosinophils all the way around them. They're much, much bigger than the eosinophil. It's not phagocytizing it. It's just the eosinophil, as you guys know, is loaded with factors that impact the tissue, impact cells around the region. And there is a benefit of that diarrhea response, that physical expulsion response within the gut, the coughing in the lungs, the runny nose, the eyes, the tearing of the eyes, that physical expulsion. And it's really factors from mast cells and eosinophils that are contributing to that. We will talk about that a lot in our hypersensitivity talk. All right, I mentioned briefly, well, actually a couple times now, the IgM, first antibody made. It's our oldest antibody when we look back on the evolutionary scale, but it's not a real effective antibody most of the time. One of the reasons why it's not effective, I mentioned, was its affinity isn't always the best for antigens. And so one mechanism that increased the effectiveness of IgM was to hook 5 of them together. And so we've got a pentameric binding structure. And so we increase avidity without impacting affinity here. It's a very simplistic way of increasing the effectiveness of the antibody. That increases its binding, but the other thing that it does is that for activation of complement, and I've mentioned complement I think 2 or 3 times now, for activation of complement, bringing antibody molecules in close proximity facilitates the activation of complement, and IgM is really good at activating complement. Complement's involved in our initial inflammatory response, so it kind of goes hand in hand. If IgM's the first one made, it also can contribute to that ongoing inflammatory response within the site. But most of the time, we're going to get a class switch to IgG in this setting, and other classes. But we'll get a class switch to IgG, and AID is involved in there as well. So I probably should have asked my question here, then it would have been an easier answer. AID is contributing to affinity maturation, better binding, and so forth, and class switching. So, in the process. All right. Any other questions on that particular? That was a good question about sequence. Yeah. Yep.
Oh, just a quick question. So do the B cells are activated outside of the lymph nodes and move there, or also
So,
are pathogens?
We'll go into more detail when we talk about that responding B cell after it's developed. But the naive mature cell that comes out of the bone marrow into the blood, in the normal scheme of things, there's always an exception to everything, but in the normal scheme of things, it's now circulating through our secondary lymphoid organs. And the secondary lymphoid organs are going to be collecting the antigen. So the bulk of the time, that initial contact with antigens taking place in the lymph node, that's really that role of that draining lymph node. The B cell comes in through those postcapillary venules and goes into the cortex, and it will be on a mission to go to the follicle. But if there's antigen in the node, and that's on our dendritic cells and our follicular dendritic cells, interdigitating cells, it's starting to interact with antigen as it's moving to the follicle. T cells are interacting with antigen, and depending upon how robust the response is, we're getting communication right away. But the B cell's moving to the follicle. And we will have a few T cells go into the follicle to provide help. But it's as that B cell is moving that it's interacting with T helper cells, dendritic cells, and so forth. Then it's moving into the germinal center. And in the germinal center, it is actively expanding. We get differentiation into plasma cells. It's a really, really hot zone. And it's in that hot zone that, whoops, I've already switched, the mutation events, and then eventually class switching and so forth takes place. But the response can start very, very early after that B cell enters into the lymph node. And we've got these cool factors that regulate all that as well that we'll be talking about. So we quickly went through the B cell receptor. The T cell receptor has many of the same common features for generating diversity. So we'll be able to just breeze through those. And so, but we have a couple key differences that we have to consider when we're talking about the generation of T cell diversity. The TCR has got kind of a weird feature that isn't the case for a B cell, is that it actually needs to be able to recognize self. It needs to know that it's your own MHC that it's going to interact with that's presenting peptide. So it has to be able to recognize your MHC. The TCR does, which is kind of a little scary, but it does. So this is unique to the TCR, that our TCR has to be able to tell if the antigen presenting cell is self. Okay? It does not recognize free antigen. The B cell can. The B cell can recognize native antigen in my early cartoons and so forth. But TCR is not. It's going to recognize peptides presented by MHC. And it's this restriction by MHC that is really unique to the T cell. The repertoire, the diversity aspect, is selected and generated in the thymus. It is in this organ that the T cell is going to learn to react to both self and non-self. but it can't react to self so strongly that it causes an autoimmune disease. And so the education in the thymus is wild. In that we need to put out this cell that's got a little ability to see our own cells, but not too tightly, and doesn't drive an active response. That's that our T cells kill our own cells. But then it's only going to recognize foreign peptide that's presented by our MHC. Okay, and so they're going to do this with the T cell receptor. The diversity, that binding pocket, many of the rules are the same. However, there are some differences, and I'll tell you the big one. The generation of diversity before antigen, very, very similar. The big difference is after antigen. TCRs never get better. Okay, affinity maturation is not due to making a better TCR after we encounter peptide. And so explain that one to me. How could we have a few T cells in that germinal center and having these mutation events occurring only in the B cell, in that B cell region? The mutations are not occurring in the follicular dendritic cell. It's not occurring in the T cell. It's occurring in those loci within the B cell. Those DNA sequences are still in the T cell. They haven't been recombined. And there, so that's pretty wild that mutation event, those somatic mutations, are so tightly controlled. They're preferentially only occurring in the B cell and only in that germinal center. T cells, which have now a recombined region and so forth, we're not going to get somatic mutations taking place. So they do not get better. So the first one we make better be good. Okay, so I've already indicated this aspect. Now for the TCR, it's a smaller molecule. We have only 2 chains to consider, but we happen to have 2 T cells that we have to talk about. One is called an alpha beta T cell, and that's just related to what we call the chains, the alpha chain or the beta chain. So we call it an alpha beta T cell. You know, 90% of what we're going to talk about in immunology and what you'll consider down the road in your patients and so forth really is in the context of alpha beta T cells. And to show you that I don't give you my bias, my research bias in this class, I spent 25 years working on the other guy. That doesn't really do anything. It might make you question my intelligence and so forth. But we have another T cell called a gamma delta T cell. And it has a receptor just like this, but the chains are different. And they're denoted gamma and delta. And gamma delta T cells have a variable region just like the alpha beta. They have some unusual features in how they can react to antigen and so forth. But the recombination events are very, very similar for the gamma delta and alpha beta T cell. But we'll spend most of our time on the alpha beta T cell. We're going to generate rearrangements, and it's the same story as far as moving gene sequences together. So I think we'll be able to go through that quickly. And the mature T cell that we're going to try to generate, naive mature T cell, in the context of the alpha beta, we're going to have one functional alpha, one functional beta. Though the T cell, because we have rearrangements occurring on a number of loci, because when the T cell is a progenitor, it doesn't know it's going to be a gamma delta. It doesn't know it's going to be an alpha beta. It's trying to rearrange both. We do have some T cells at the end of the day that have rearranged both. Okay, I told you the rules in the B cell are pretty clear. We just get one successful recombination, and we have that allele exclusion. In T cells, we can find rare T cells that have recombined multiple loci. Okay, so that's another difference for this population. The diversity that we're going to generate, we still have CDRs, 1, 2, 3. It's the same concept of the loops and so forth that are going to contribute to binding regions, and the nomenclature is the same. We know a lot about crystal structures and the structural formation of the binding pockets and so forth. We have regions, so constant domains and variable regions. Constant domains are making sure the three-dimensional presentation of the variable region is there. And we know all of that from detailed X-ray crystallography and such. The recombination events, the story is the same. I don't think I have to repeat everything. We have loci for the alpha, and we have loci for the beta, and they have different copies of gene sequences. Variable, indicated in red here, and joining, indicated in yellow here. And the alpha chain is like the light chain. We have just those two, multiple copies of each one. They're going to be brought together. And the beta chain is like our heavy chain. We've got another region, that D region, in there. So at the end of the day, we recombine and we bring two regions here, a V and a J region, together in our alpha chain. In our beta chain, we're going to be putting in a D region in there as well. So that should be similar if you go back and look at the rearrangements in the light chains of an antibody versus the heavy chain. So that mechanism was conserved, that aspect was conserved. And we're going to make a transmembrane form, and we do not have any issues with RNA splicing. And again, as I try to simplify this, not to go on and on and on about recombination, so I just have these bullets just to make sure that you remember. The diversity that's impacted by the addition of P and N nucleotides, that occurs as well in this setting. So we're having these recombination events occurring in our alpha and our beta. They occur sequentially, and I'll spend more time talking about the sequence when we talk about T cell development. And then we're also having rearrangements occurring on our gamma and delta. And it's when the T cell is learning to be a T cell, this is happening. Rearrangements, rearrangements, rearrangements. And then when we get a successful transcript, successful protein, then we move forward. And the cell just keeps trying. And if never can succeed, it just eventually dies. So those RAG proteins, remember those? Control that recombination event. Same thing in the context of T cells. So we do have lectures in this course, both WAMI and the WEMU students, that are going to be focused on immunodeficiency diseases. It's always good to throw a little clinical tangent in. And somebody earlier, when I was asking about what would be a phenotype if you lacked AID, I think that was the context of that question, or one other enzyme, had mentioned SCID. So SCID is an acronym for Severe Combined Immunodeficiency. And this is where your patient, be it an animal or a human, is lacking, in most instances, complete B and T responses. When I say most, not necessarily all. You can have a SCID phenotype and still have a little antibody being made. But for the most part, we have no cellular mediated immunity, and it's either no antibody or only a little antibody that's being produced. This occurs and has been seen virtually in every animal that has an immune system that undergoes recombination. And so the bad SCIDs are the ones that you're lacking enzymes or proteins involved in the recombination event. Go back to the B cell lecture. I gave you the one slide of the enzymes involved in opening up that hairpin loop to bring the pieces together and then adding the N nucleotides and so forth. If you lack those enzymes, you can't do that. That's going to end up being a SCID. If you lack RAG, you can't do recombination, end up being a SCID. So we knew this phenotype existed in humans first in the context just because we had the detailed diagnostic tools, but the insight in molecular mechanisms and so forth came from identifying the same phenotype in animal models. And one of the first ones actually was in Arabian horses. This was actually done at WSU. I was actually getting my PhD at the time when this data was coming out. And in these horses, just like in a human patient, they actually lack functional B and T cells. And the genetics behind this is the same as defined in horses as in humans. They're normally autosomal recessive traits. And so you need both copies of the gene to have the deficiency. Or, excuse me, both copies of the mutated gene to lead to the deficiency. And so in horses, these foals die within 4 to 5 months of age. Why does it take that long? Yeah. Yeah. That's another thing that's going to mess up your diagnosis when you're dealing with an infant that, it could be healthy, and all of a sudden at 6 months, they're coming down, here's another bacterial infection, here's another one, antibiotics and so forth. And the 6 months, the maternal passive immunity that was transferred was holding things in check. Innate immunity is just fine. But you'll start to see problems, later on. And when it's a severe, this is where you're seeing bacterial, intracellular, extracellular. You're seeing viruses. You're seeing, fungal infections, maybe some parasitic infections and so forth. When you're seeing that in your patient, boy, be looking immediately at severe combined immunodeficiency. That they're lacking. The first thing I would be looking at, they're just lacking the mechanisms for these recombination events. So they're not generating the repertoire. So they're born, they're maintained early in life because of passive immunity from the mother. But then when they need to be able to react to that foreign protein that comes in, they have no receptor. They have no antibody, they have no T cell receptors in that setting. And so repeated infections, lymphocyte numbers, they're not gone, but they're greatly reduced. And we're going to be talking about why lymphocytes aren't necessarily gone in these patients. And one reason is we have these oddball lymphocytes. One of them is called this NK cell, I think I mentioned in the first hour today. And in some instances, their numbers can be much higher than a healthy individual. And so you do see lymphocytes. You're not seeing B and T cells with rearranged receptors. And that's the key thing that you're looking at. Lymph nodes will be small, thymus will be small, lack of antibodies. And if you don't have IgM, you didn't have a B cell. Okay? If you see lots of IgM, you had a B cell. But if they didn't have IgG, there was something to deal with, class switching or something along those lines. And so, this is just in the context of kind of the story. We see these in certain livestock animals, that certain genetic deficiencies, and sometimes we actually select for them. And in this particular context, in the Arabian population, they were able to track it back to a single stallion that was imported in the 1920s. There's another story that's relevant to both human and veterinary medicine of a human immunodeficiency. And this is a deficiency in an important molecule involved in inflammation. And it's how the cell in the blood sticks to the endothelium. And there's a molecule that's called an integrin that's involved in that binding. And back in the late 80s, there were human patients that were identified that neutrophils weren't seen in their inflammatory lesions, and they very severe immunodeficiency. It's not a skid, but it's severe because it impacted innate responses. So neutrophils couldn't get in to control infections. And this phenotype comes up very early in your patient. And because the baby is going to be being exposed just to common bacteria and so forth, and the leukocytes can't get out of the blood. and get to the site to fight off the infection. It's called leukocyte adhesion deficiency. There's different forms of it. It was defined in the human at the genetic level, but we really didn't have animal models to be able to go in and actually prove mechanism, so forth, except in the dairy industry. A unique story came out that in dairy cattle, for production, one of the key defining things in which you're either going to pull that cow out of production or keep it in is the somatic cell count in milk. So how many inflammatory cells in milk? That's the key. When it gets too high, then you pull that animal out. So you select for animals that are more resistant to infection, and you don't see inflammatory cells in the milk as often. Well, this particular immunodeficiency is not autosomal recessive. It's actually, if you're a heterozygote, you have half the adhesion molecules, half the inflammatory lesion. So what they ended up doing in the U.S., this occurred, was selecting for cattle that had lower inflammation rates because the heterozygotes were half the inflammatory cells. So our milk still had the same bugs and everything in it, we just had lower cell counts in there. And it was a combination of MDs working with DVMs that identified this, and it was all due to selection for this leukocyte adhesion deficiency in cattle. Now, the cattle that had both defective genes, it then was a severe deficiency. So you guys will get this and subsequent lectures on inflammation and the impact of loss of the ability of the cell to get out of the vascular system. So a little tangent there. I just put this back up just to remind you. So if we lose this enzyme, so we can't clip, we got to open up these hairpin loops so that we can start sticking things on. We can't do the process. You got loss of recombination. TDT, loss of recombination if you lack that. DNA-dependent protein kinase, and that's actually what they found in the Arabian foals, accounted for their lack of recombination. In humans, it's going to come up quick, and a common presentation, obviously, if you lack RAG, loss of B and T cells, but in the infant, you're seeing bugs that you just shouldn't be seeing. And one of the common ones is a fungus, Candida albicans. This is really kind of the smoking gun in so many instances when you see recurrent Candida albicans infections that there is an impact on host immunity in that context. And for a SCID, for lacking B and T cells, it happens really early. The passive immunity from the mother can't really deal with the bugs in this setting. It will pop up early when it's severe like this and lack of RAG. Okay, back from the clinical tangent to the T cell. You guys have heard me use CD now over and over, CD4, CD8, so forth. The CD comes from just our attempt years ago to try to reduce the minutia in immunology, because in the old days, if we discovered a receptor, we just called it whatever we wanted. And if it was a receptor for growth, we called it T cell growth factor receptor. If it was a receptor for something else, we tried to link it to the function. But it really made a quagmire, because I would give it a name and somebody else would give it a different name, and so the literature was really, really confusing. And so we came up with a common designation, this cluster of differentiation antigens, CD. So the molecules on our immune cells are antigens. If we stick them into the human cell into a mouse, the mouse is going to make immune responses for it. We call those things CD. So CD4 is a molecule on a T cell, a T helper cell. CD8's a molecule, and so we're going to be using this nomenclature over and over. That's where the CD comes from. So for the TCR, when we make a functional TCR, so we've recombined the variable region, and we've generated diversity in the CD3, most of the diversity from the junctional P and N nucleotides. We make this TCR, and remember in the B cell lecture, we express it on the cell surface, but it quickly associates with other proteins. Just like that picture for the surface IG associated with other proteins. These other proteins are involved in signals that can be delivered to the cell when the TCR binds to something. And this is where the nomenclature just adds a level of complexity. We have TCR associating with these other proteins, and this complex is called the CD3 complex. An important concept. We can express TCR, but if it's in the absence of these accessory proteins and we don't have the complex, the cell isn't going to receive the appropriate signals. And so you will see questions and concepts in immunology when they're talking about the T cell. Make sure you're looking at those issues of whether or not they're simply talking about antigen recognition or signaling, or both. You cannot get signaling without this complex. So for the real McCoy, the real T cell that needs to keep your patient healthy, when they express a TCR, they're always going to express the CD3 complex. They go hand in hand. And if they don't go hand in hand, you have a problem. So the functional complex is what we need to always be considering. And this is the CD3. This will drive a signaling response that we'll talk about later. And for the B cell, I gave you the SYK kinase as being involved in a signaling response that's unique to the B cell. For the T cell that we will talk about, and I expect you to remember ZAP70, I'll bring it up again. We actually have deficiencies in ZAP70, and it'll present in a unique fashion in your patient. And again, I mentioned with the B cell, so we have this complex that's going to see antigen here, and we're going to drive signals to tell our cell what to do. We also will have other receptors out here that we'll talk about later, co-receptors that are involved in really driving the appropriate response of the cell. So it does get more and more complex as we put it all together. But right now, we're just talking about the T cell response, the recombination events that take place. I told you it's also happening on this oddball cell, this gamma delta T cell. Alpha beta T cells are the predominant one in the human. If I took a sample of your blood, looked at the frequency, it's gamma delta T cells might be 5%, 3%. So they tended to be ignored in the human. Not so much these days, but pretty much through the 90s and so forth, where a lot more effort was put as far as a focus on their role in host defense or in certain animals such as cattle. 60-70% of the T cells can be gamma delta T cells in that setting. So we have differences between animal species and so forth, but every single animal that has a complex immune system have these cells, and they were the first to arrive in evolution. They actually came up before the alpha beta, and they tend to be their functions aren't as restricted. They're involved in a lot of tissue functions, and we don't really bring these cells into the equation when we talk about cytolytic cells and helper cells and so forth. These cells tend to be able to do whatever they want under whatever condition they may be found in and so forth. But they are rearranging their TCRs. The cells, those loci are rearranging just when the alpha beta are rearranging. And so we are getting rearrangement events, and it's kind of a race to see who's going to come out first, an alpha beta or a gamma delta. As MHC is going to allow the T cell to distinguish self and non-self, and it needs to know that the antigen presenting cell is self. And the only way to do that is that it has to recognize it. And so, you know, here we have this T cell, particularly the story with the cytolytic T cell, with the potential to be able to kill. It's got to be able to see our own cell, and it's using its TCR to do that. But it's only going to lead to an eventual cytolytic response if there's foreign peptide there. When we do grafts, the big problem in a graft rejection process is that when we mix two different MHCs, that just triggers overt responses, not normal, overt responses. Yeah?
So this says that the person suffers from most nucleated cells. Does that mean red blood cells do not have any?
Yeah, so I think I made the statement that MHC class 1 in my overview is expressed on nearly all cells. The exceptions for MHC class 1 are red blood cells, cells in the brain, nerve cells and so forth, and some other privileged sites. And certain pathogens, for example, take advantage of that. So the herpes virus, it takes advantage of hiding out in cells that have very, very low levels of MHC class 1. So nerve cells. And, you know, if you get cold sores, the reason why you never get rid of that virus is that when it goes into its latent state, it's sitting in a cell with very, very low levels of MHC class 1. So you have great cytolytic T cells that can kill it, kill the host cell, but if the host cell doesn't have MHC class 1, you can't get to it. So it sits there, and then when you're stressed in immunology and factors occur, the steroids are released and so forth, that virus reactivates, comes down the nerve, hits the epithelial cells. It loves epithelial cells. It's generating progeny. You've got CTLs that just fry it, and that's the cold sore. That's occurring. Then you get over the cold sore, the virus goes back up into its state in the cell that doesn't have much MHC class 1. So certain bugs have found those cells and they take advantage of it. And then other viruses, what they do is they go into our normal cells and downregulate the MHC class 1. There's another competitive advantage in that context. So we'll end here, and this is what we're going to transition to in finishing this discussion of how a TCR is going to recognize foreign peptide, and then talk about MHC restriction and actually differences between us that you guys have to consider in transplants and in other scenarios and so forth. All right.